Osmosis is the flow of water across the membrane, and the water will flow from where the water concentration is highest to where the water concentration is lowest. So inside the cell, there are a lot of solutes. If you put that cell into water, water will flow into the cell because there's a higher solute concentration inside the cell. Osmotic flow is the movement of water that's driven by osmosis, and osmotic pressure is a force. It's the force of water moving into the solution with a higher solute concentration. This diagram represents osmosis. Here you have a tube, and that dashed line represents a membrane, okay? And it starts out with more water molecules on the left, the small blue dots, and more solute molecules on the right. Water tends to flow from high concentration, so on the left here you have a higher concentration of water, so water will flow from the left side to the right side. You can look at it also as using this phrase, water follows salt. Salt being any solute. So water flows towards where there's a higher salt or solute concentration. Now over here on the right, if you were to push against the fluid, you would generate a hydrostatic pressure. The osmotic pressure in solution B is equal to the amount of hydrostatic pressure, and that's indicated by this weight here. So the force applied to this fluid, this volume B here, to equal the force, to equalize the force of A and B, this force here is the hydrostatic pressure. Don't worry about osmolarity, it's just the total solute concentration. Often we look at percentage, like our blood has 0.9% salt solution. Tonicity is an important concept. We'll look at three types of tonicity: hypotonic, hypertonic, and isotonic. And I think you know at this point that hypo means low, hyper means above or high, and iso means the same. So these pictures show red blood cells placed in different solutions. In the middle, isotonic, the red blood cells look normal. They have been placed in a solution that is isotonic with the cell. That is, the solute solution outside the cell is the same as the solute solution inside the cell. We see this when we are in the hospital and we have to have an IV solution. They will never give us pure water. They will give us a solution that is isotonic with our blood and our blood cells, because you don't want a net movement of water into or out of the cell. On the left-hand side here, hypertonic, these cells have been put into a hypertonic solution. That is, they have been put into a high salt solution, higher than blood. Blood is 0.9% sodium chloride. So let's say they put these blood cells into a 10% salt solution. The water will flow out of the cells, and the cells will crenulate. And you can see here that these cells have shrunk because the water has flown out of the cells. So water has gone to where there's a higher salt solution out of the cell. Water follows salt. And over here on the right in the hypotonic solution, and distilled water or plain water is always the hypotonic solution. So here red blood cells have been put into a pure water solution. There are more solutes in the cell, so water flows into the cells, and the cells burst. This is hemolysis. The importance of tonicity and osmolarity. Again, osmolarity is really just the concentration of solute. So when you administer large volumes of fluid to patients with blood loss, those fluids have to have the same tonicity as their blood. So there is no net flow of water into or out of the cells. A 0.9% sodium chloride solution is isotonic with the blood. Another type of transport is carrier-mediated. Our membranes have proteins in them, and these carrier proteins will help nutrients and wastes be transported across that phospholipid bilayer in our membrane. Many of these carrier transport proteins move molecules only in one direction. Countertransport is moving in two molecules in opposite directions. These are called exchange pumps, and the main one we talk about is the sodium-potassium pump, where 3 sodium ions are pumped out of the cell at the same time that 2 potassium ions are pumped into the cell. So 3 types of carrier-mediated transport. Diffusion, facilitated diffusion is the first one. Remember, diffusion is passive. It does not require ATP. And here we have a carrier molecule that's open, and it's allowing molecules to move from high concentration to low concentration. Therefore, it's diffusion. Active transport requires ATP, and examples of these are ion pumps. Now, it says here independent of concentration gradient. So what's happening is let's say the cell requires magnesium, it's going to pump magnesium into the cell, regardless of the concentrations outside or into the cell. And then our sodium-potassium pump is our key exchange pump. Facilitated diffusion, again, is just no ATP is required, and you have a specific molecule, such as glucose in this case, binds to a receptor site on the integral protein. And integral just means that protein spans the entire membrane. And then the shape of the protein changes, allowing the glucose molecule into the cell. Active transport, here we're showing the sodium-potassium pump, which is our key exchange pump. Sodium concentrations are always high outside the cell. Think of your cells as being in salt water, and sodium constantly leaks into the cell. This is not good for the cell, so the cell pumps the sodium out. On the other hand, the cell requires higher concentrations of potassium, so it is pumping potassium in as it pumps the sodium out. 2 potassium in for every 3 sodium out. This requires ATP. Carrier-mediated transport, secondary active transport. We don't really, we don't talk about this much. Just read through this. But again, it's active transport, and therefore ATP is required. Vesicular transport is pretty easy. Membranes, the membrane forms a small vesicle to bring substances in. A very actively transporting cell will replace its entire membrane through this vesicular transport in about an hour. 2 major types: endocytosis, bringing products into the cell; exocytosis, releasing products from the cell, and both of them are active, requiring ATP. Endocytosis may require a receptor. This would be receptor-mediated endocytosis. Now, a ligand is just a molecule. So the molecule binds to the receptor. There are many receptors in a certain area of the membrane, and it kind of folds in or invaginates, forming a vesicle. the vesicle will fuse with a lysosome, and the lysosome will allow these molecules to be freed and enter the cell. This whole process, you can pause the video here and read through this entire process. Endocytosis is divided into two categories. One is cell drinking, where liquids or small particles are brought in. Phagocytosis is cell eating, so it would be larger particles such as a white blood cell or macrophage engulfing a bacterial cell. Exocytosis is discharging materials into the extracellular fluid. This picture shows it really nicely. You can see these invaginations here of bringing in, this groove here is formed and a vesicle develops. The important thing about this phagocytosis is the object that's brought in, of course, must be inside a vesicle, because when the lysosomes fuse with that vesicle, you don't want the lysosomal enzymes released into the cell. That would cause the cell to die, or autolysis, perhaps you remember that term. The cell life cycle is important in that every cell undergoes its own life cycle. They will have life cycles of different time periods. Cells divide by two processes. One that we talk about the most in A&P is mitosis, where one cell becomes two cells, and the two daughter cells will have exactly the same chromosomes as the parent cell. Meiosis occurs to form sex cells. It occurs in the gonads, the ovaries and testicles, and those cells will only have half the chromosome number. Again, mitosis results in two daughter cells that have identical chromosomes. At the end of mitosis, the cytoplasm divides, and that's called cytokinesis. And then the cell is going to just do what the cells do. They're going to enter interphase, which is the majority of the life of the cell. Probably 23 out of 24 hours would be interphase, and they do what they do. A pancreatic cell would be producing and secreting insulin. Some cells would be making mucus. Lung cells would be diffusing gases. So in interphase, that is the life of the cell between divisions, there are different stages of it, mainly G1, S, and G2. Now G0 is a stage a cell will enter when it never divides. So it goes into G0 and just stays there. It may, if for a skeletal muscle cell, it may stay there unless it is damaged, and then it will exit out of G0 and go on to repair itself and divide. Stem cells never enter G0, they constantly divide. G1 is the normal cell function, but the cell is going to grow, it's going to increase the number of organelles. In the S phase, the chromosomes are copied or duplicated. And in G2, before mitosis occurs, certain proteins have to be made, specifically microtubules, and the centriole has to replicate for cell division to occur properly.